Hello class, this is Dr. Anderson, and this is the first in a series of 3 presentations on metabolism. First, we're going to start with the absorptive state. Here are our learning objectives. So you've just talked about the gastrointestinal system and the absorption of glucose, amino acids, and fats. So one of the things we need to talk about is that glucose and those amino acids that are taken up into the hepatic portal vein, they go to the liver. We want to think about what the liver's doing with those substrates, and then when those substrates, the liver removes some substrates, and then they go on to the rest of the body and they get used and stored. Now, your liver during the absorptive state perform some processes. First is called glycogenesis. That is the process of taking glucose molecules and covalently binding them together to form glycogen. And your liver also performs something called lipogenesis, which is taking substrates, turning them into fatty acids and glycerol, and then turning that into triglyceride that gets packaged in a lipoprotein called the VLDL, and you put that into the blood and send it to the adipose tissue. And what I would really like the class to appreciate is that because the liver gets the first crack at the glucose coming from the digestive tract, much of that glucose is stored as glycogen and fat, and then the rest is sent on to the rest of the body. So the glucose level that the rest of the body experiences is not nearly as high as it would be if that glucose were just sent straight out to the body. So the liver acts like a buffer for the rest of the body to prevent these big, large spikes in plasma glucose. We also want to think about the idea that you ingest some fatty acids, triglycerides that are broken down into glycerol and fatty acids, reassembled into triglycerides, packaged in a chylomicron, and sent on to the adipose tissue. But your liver also can make new fats, and those fats are packaged in VLDLs and sent to adipose tissue. So how do we regulate what kind of things are going on in the post-absorptive state? Well, we need a very important hormone, and that's called insulin. And insulin is regulated by negative feedback regulation, and the most important parameter being regulated is blood glucose level. If your blood glucose level goes up too high, then you secrete insulin. And insulin is going to help you get glucose into your cells and help you store all the substances you ate during the absorptive state. And we're going to finish with thinking about the connection between diabetes and hyperglycemia. Once you figure out what insulin does to regulate your blood glucose level, then students ought to predict, well, if you're not producing insulin or not responding to the insulin that you have, you're not going to get the maximal effects from glucose or from insulin. And what is that going to mean for your blood glucose level? Alrighty, so our metabolism very much depends on how long it's been since you've had a meal. So in the absorptive state, you just ate a meal. And many times we assume that you've eaten a balanced meal. Not always do we eat a balanced meal. But as we discuss these concepts, we're going to sort of pretend like you had a balanced meal. So you have all these nutrients that go in the bloodstream from the GI tract, and they go to the liver. And so your body is in a storage state. Then later on, and typically it's about 4 hours after you've had a meal, that's sort of the average amount of time that goes by. Well, you need to do things in order to maintain your blood glucose level, and you're going to go to all those nutrients that you stored, and you're going to break those down to make sure the body has nutrients. So in this presentation, we are going to discuss the absorptive state. So in general, the post-absorptive state is when you've had a meal and those about 4 hours after. And the predominant kind of metabolism is a process called anabolism. Anabolism basically is building reactions. You take fatty acid and glycerol and you put them together to form a more complicated molecule called a triglyceride. You take amino acids and you covalently bond them together to form a more complicated molecule called a peptide or a protein. You take glucose molecules and you covalently bond them together and you form a more complicated molecule called glycogen. So storage reactions are the predominant type of metabolism occurring right after a meal. Since you've just eaten a meal and there's plenty of glucose, that's what your body's going to run on. Most cells of your body are going to use glucose to make ATP. Extra glucose is going to be stored as glycogen and fat, and the liver is going to package that fat and send it to adipose tissue. And the most important hormone during this time is insulin. Insulin, when it binds to its receptors, is going to promote the reactions of the absorptive state. So what kind of reactions do we mean? Well, I told you that the most abundant reaction are anabolism, but there are going to be a couple of kinds of catabolism. One is the catabolism of glucose. Glucose is taken up into cells, it gets phosphorylated, and that is the first step of a series of reactions we call glycolysis. Now, some of you may be majoring in biochemistry and you think of glycolysis in a different way, but I basically think of you have a 6-carbon molecule, you phosphorylate it to destabilize the molecule, you break it in half, and you release some energy. So at the end of glycolysis, you have 2 3-carbon molecules called pyruvate. You are going to be breaking down some amino acids for use by the liver. And to use an amino acid, you have to take the amine group off, and that's a process called deamination. And when you deaminate an amino acid, that amino group becomes ammonia. Now, ammonia is toxic, and you can't have that running around your body. So your liver takes the ammonia and turns it into a less toxic form called urea. And I'm sure you heard about the molecule urea during the kidney portion of this course. So urea is a substance that makes the, something happened here, that makes the medullary interstitium of the kidney very, very concentrated, along with sodium chloride. So urea comes from the deamination of amino acids. Now, many of amino acids are not going to be deaminated. They're going to be linked together to make proteins. So protein synthesis is a reaction that you will observe during the absorptive state. Maybe you'll break down a few lipids. However, this next reaction, glycogenesis. Glycogenesis is the process of glycogen synthesis. So the liver and skeletal muscle are the 2 places where glycogenesis is going to take place. That's basically taking glucose molecules and stringing them along into long molecules called glycogen. Then lipogenesis is a very important reaction of the absorptive state. Lipogenesis is going to occur in the liver and in adipose tissue or fat tissue. And remember, a triglyceride is a 3-carbon glycerol backbone that is covalently bound to 3 fatty acids. And there are longer chain fatty acids, and they usually have an even number of carbons, so 12 carbons or 13 carbons or 14 carbons or on and on and on. And that is what makes a triglyceride. The triglycerides that are made in the liver then are packaged in a VLDL and sent to adipose tissue. All right. So what happens to the food that you eat? Glucose and amino acids are absorbed into the capillaries of the villus. Remember, they cross the luminal side of the cell, then the basolateral side of the cell, and they move into the capillaries. That glucose and amino acids, they travel to the liver by the way of the hepatic portal vein. Also remember that there was a process of emulsification and fat digestion. Those are absorbed with the help of my cells, and inside the intestinal cells, those that glycerol and those fatty acids are turned back into triglycerides. They're packaged into a chylomicron. The chylomicron is exocytosed out of the cell, and because chylomicrons are so large, they cannot go into a capillary. They have to go into a lymph lacteal. So that fat travels around in the lymphatic system, and eventually the lymphatics empty from the thoracic duct into the venous system. They circulate throughout the venous system and eventually are going to make their way to the arterial system so that they can be delivered to adipose tissue. So here's a very nice diagram, and I think I will get out my little Tinkerbell so you can see all the things going on. All righty. We have just eaten a meal. So there are glucose in the blood, amino acids in the blood, and some triglycerides that have entered the blood by the way of the chylomicrons in the lymph. All right. So we had a meal, and now we have substrates in the blood. First and foremost, in the absorptive state, most tissues of the body use glucose. Glucose is taken up into the cell. It's burned through glycolysis, the Krebs cycle, and oxidative phosphorylation. All the carbons in glucose eventually become CO2. The oxygen picks up the electrons from the electron transport chain and becomes water, and the energy in the bonds becomes ATP. So most cells of the body are running on glucose. Glucose also makes its way into the liver. The liver takes that glucose and synthesizes it into glycogen. That process is called glycogenolysis. However, the liver is also very good at taking excess glucose and turning that glucose into glyceraldehyde, or a phosphorylated version of glycerol, and taking that glucose and turning it into fatty acids. Glycerol and 3 fatty acids are then turned into a triglyceride and packaged in a VLDL. Also notice in the liver there are some reactions going on with amino acids. So amino acids enter the liver. The liver can use those amino acids to make proteins. Think of a very important protein your liver makes. Did you all come up with albumin? Good job if you remembered albumin. And the liver makes a whole lot of other proteins. It makes all of your clotting proteins. It makes the proteins that are part of the complement cascade. Maybe if you've taken cell biology, you've learned about the complement cascade as a part of defense. So important proteins are made by the liver. The liver itself does not run on glucose. It's a very kind of a weird thing, but the liver's job is to process glucose. So it doesn't use glucose itself. It uses a molecule called a keto acid. And a keto acid is basically a deaminated amino acid. So then that keto acid can be used by the liver to make ATP, but keto acids also can be used to make fatty acids and triglycerides. So the liver is storing glucose as glycogen, storing amino acids as proteins, using keto acids as energy, and taking the excess, storing it as triglycerides, packaging them as VLDLs, which are then sent to adipose tissue. Now there's an interesting process that fat has to go through. When these triglycerides are delivered to adipose tissue, there's a very important enzyme in the capillaries. It's called lipoprotein lipase. And it basically takes the triglycerides, breaks them down into a monoglyceride and free fatty acids. Those diffuse into the adipose cells, and then they get turned back into triglycerides. Now, why go through all the trouble of that? Well, remember, triglycerides are huge. They can't just diffuse into a cell. You have to take them apart, let the little smaller parts diffuse into the cell, and then you put them back into triglycerides. Now notice that adipose tissue also receives glucose. That glucose goes into the adipose tissue, and glucose is going to burn, adipose cells are going to burn glucose for energy. They're going to take the excess and turn it into fat. Our bodies are very, very good at making fat. It kind of makes me mad, but it's very, very easy for your body to synthesize fat. And I think that's because this system was evolved under conditions where people and animals didn't necessarily have access to food all the time. So they needed to be able to store all the food they ate and save it for later when there was no food. And that may not be the kind of life we're living now. And so, which, you know, we have an obesity crisis in this country. And so that's because we live in a different world as compared to the metabolism that we have. All right. We've talked about most cells of the body. We've talked about the liver. We've talked about adipose tissue. We also have skeletal muscle. Skeletal muscle is going to take up the glucose, and skeletal muscle will use the glucose to make ATP and use the glucose to make glycogen. In addition, amino acids are going to go into the muscle, and the muscle is going to use those amino acids to make protein. So all your actin and your myosin and your tropomyosin and your troponin and all of your intracellular enzymes. Muscle's a very protein-intensive cell, and it's constantly repairing old, worn-out proteins and putting in new proteins. So there's an amino acid requirement for your muscle cells. So I've taken some time to go through that diagram, and for those of you that maybe like words better than pictures, I'm going to go through the whole thing again, only with words instead of just pointing to a picture. So where do we start? Most cells of the body are using glucose. So glucose enters most cells via a GLUT transporter. Some of the GLUT transporters need insulin to be there. So those are called insulin-dependent cells, and muscle is a great example of an insulin-dependent cell. Some cells are insulin-independent, meaning the GLUT transporters are there all the time, regardless of whether you have insulin or not. And the brain is a great example of an insulin-independent tissue. So the brain can use glucose anytime it wants. Now, as soon as glucose enters the cell, it gets phosphorylated. That's the first step of glycolysis. So a couple of things happen. You get glucose into the cell, you phosphorylate it and get ready to get it into glycolysis. But if you phosphorylate the glucose, now it's glucose 6-phosphate. It's not glucose anymore. And you maintain the inward concentration for the facilitated diffusion of glucose. So there are a couple of buzzwords we use. We talk about glucose uptake and utilization. What the heck does that mean? Well, if glucose gets into the cell, then that's uptake. And if the cell uses the energy in glucose in glycolysis, that's utilization. All right, now, the liver is very important in this process. The liver does not use glucose itself for energy. It uses deaminated amino acids. And so when you take the amino group off, remember you have that ammonia that has to be detoxified and turned into urea, but the carbon portion you have left is called an alpha keto acid. So the liver uses that for energy. The liver also can make fat out of that. The liver takes those ingested amino acids and turns them into protein. Remember the examples I gave you? Albumin, clotting proteins, so thrombin and fibrin and all of those guys, elements of the complement cascade. There's some binding proteins that your liver makes, so very, very important. The liver uses glucose that was uptaken after the meal to synthesize and store glycogen. And any excess glucose and amino acids, for that matter, those are converted into fatty acid and glycerol. Those are made into a triglyceride, packaged as a lipoprotein called a VLDL. VLDL stands for very low density lipoprotein, which means this is a lipoprotein that's mostly made of lipid. So it is a low density molecule. Those enable the triglyceride to dissolve in the blood so they can be sent to adipose tissue. All right, now one of our major learning objectives. How does the liver prevent large spikes in blood glucose during the absorptive state? All right, why don't you take a piece of paper and jot down your ideas, and I'll give you a minute to think. All right, now, all the glucose that you absorbed from the meal, where's it going to go? It's going to go into the hepatic portal vein. All the glucose you eat first is sent to the liver. What does the liver do with that glucose? It takes some of the glucose and makes it into glycogen. It takes some of the glucose and makes it into fat. It sends the rest of the glucose onto the body. So the amount of glucose leaving the liver is not as high as the amount of glucose entering the liver. So the liver prevents a large spike. Now, blood glucose level is still going to go up, but not like it would were not the liver there to sort of buffer the body against a big glucose insult. All right, now what is the adipose tissue doing? The job of adipose tissue is to store fat. Now there are a group of people that don't have adipose tissue. And you might think, wouldn't that be great to be thin and not have adipose tissue? And it's actually not a great thing at all. Yes, these people are very, very thin, but they're very, very ill. And what ends up happening is they still make fat, but because they don't have adipose tissue, they store it in their muscle or their liver or various places, and it causes them some health issues. So, you know, don't be mad at your fat tissue. It's doing what it's supposed to do. You are much healthier if you store your fat in your adipose tissue. Now, if you have too much adipose tissue, does it impact your health? Certainly it does. But having some fat is not a bad thing. It's just having a healthy amount of fat. All right. Now, during the absorptive state, adipocytes or fat cells will use glucose to make ATP. They will, and they'll take extra glucose and they'll turn it into fat. They will also receive fat from the meal. So there were VLDLs that were made in the liver, and there were chylomicrons that were the ingested fat, and both these sources of fat will then be deposited into the adipose tissue. Now remember how the fat got in the cells? Had to be broken down. So in the capillaries of adipose tissue is a special enzyme that's regulated by insulin called lipoprotein lipase. It breaks down a triglyceride into a monoglyceride and 2 free fatty acids. Those components can diffuse into the adipocyte, and the adipocyte then resynthesizes it into triglyceride. And triglyceride is a good way to store energy because it's very energy dense. If you stored all of your energy as glycogen, you'd be too heavy to move around. So it's very energy dense in a lighter form than glycogen. Okay, here's an important learning objective. How is the transport of and I think I meant to say and storage of ingested fat different from the transport and storage of fat made from excess glucose? Okay, once again, I'm going to give you a few seconds to maybe jot down a few ideas. All right. When you ingest fat, you package it in a chylomicron. Chylomicron goes to lymph, then it goes to blood, and then the chylomicrons deliver fat to the adipose tissue. Synthesized fat is synthesized in the liver, made into fat, packaged in a VLDL, and then the VLDL takes it from the liver to the adipose tissue. In addition, adipose tissue can synthesize fat out of excess glucose. So the major point here is ingested fat is transported around as a chylomicron. Synthesized fat is transported as a VLDL. Okay, so muscle will use glucose for its own energy needs, especially the white, fast oxidative muscle. Some red muscle, oh shoot, what am I thinking of? And I just realized I think I wanted this to be glycolytic. I'm going to have to change that in the presentation, even though I may not get it changed in the recording. So your fast twitch muscle really needs glucose, but you have some small red oxidative fibers. That's the slow twitch muscle. Those guys can run on some fat at rest. So those muscles can use some fat, but your fast twitch muscle is going to need glucose. And not only that, your muscles are able to take glucose and store it as glycogen. That glycogen isn't going to benefit the rest of the body directly, but that glycogen is a source of energy for muscles when they're exercising. Okay, let's look back at this diagram and see if it's making sense. Remember what most tissues are doing? Most tissues are using glucose for energy. What's the liver doing? It is using keto acids for energy, making proteins, making glycogen, and excess packaged as fat and sent to the adipose tissue as a VLDL. What's triglyceride doing? It's receiving triglyceride VLDLs from the liver, triglyceride chylomicrons from the gut, and glucose for its own energy needs. Excess glucose is stored as fat. What's the muscle doing? The muscle's taking up amino acids and making proteins, taking up glucose and making its own ATP and forming glycogen. So how do we regulate this? Well, we have a beautiful enzyme called insulin. So to get glucose in most cells of the body, you need insulin. Now, there are a few cells that don't need insulin, and the GLUT transporter in hepatocytes are kind of interesting. The GLUT transporters are already there, and when you're in the absorptive state, that GLUT transporter takes glucose into the liver, and when you're in the post-absorptive state, or when you haven't eaten for a while, that same GLUT transporter allows glucose to leave the liver, but we'll talk about that on another day. Getting amino acids into the cells, insulin helps us do that. The liver and muscle to make glycogen, insulin helps us do that. And protein synthesis, now this is really complicated. Insulin, thyroid hormones, and insulin-like growth factors. So remember, insulin is permissive for growth hormone, and growth hormone causes the production of insulin-like growth factors. And then making fat in adipose cells and hepatocytes, Insulin helps us with that. So where the heck does insulin come from? Well, remember, we have this organ called the pancreas, and the pancreas produces a bunch of enzymes and produces bicarbonate. That's the exocrine pancreas. Now we want to talk about the endocrine pancreas. And in the endocrine pancreas are groups of cells clumped together in what's called an islet. And the major cell in a pancreatic islet is called a beta cell. And the beta cells are the cells that make insulin. So this is a flow chart that's not from your textbook, but I was hoping that you would find it helpful. So look at all the things that help us make insulin. If our blood glucose goes up, we secrete insulin. If our plasma amino acids go up, we secrete insulin. If we have glucose in our gut, we produce hormones called incretins. Example here is GIP, that stimulate insulin. And if we're eating and stimulate the parasympathetic system, we're stimulating insulin. Look what inhibits insulin: sympathetics and plasma epinephrine. Now, there are some feed-forward mechanisms and some negative feedback mechanisms. So here are the feed-forward mechanisms. Just by eating a meal increases the parasympathetic stimulation. You eat a meal, you chew the meal, you're smelling the meal, it's stretching your stomach, you're getting parasympathetic stimulation, and insulin secretion increases. And when you get glucose in the small intestine, you secrete a couple of incretins, GLP and GIP. These are hormones that you learned about under the GI system. They go to the pancreas and you get insulin secretion. Now, why is this feed forward? It's because parasympathetic stimulation and the incretins anticipate an increase in plasma glucose. You see, glucose in the blood has not gone up yet. The food is in your GI tract. The sugar is in your small intestine. It's not in your blood yet. But these are signals that are feeding forward and telling the pancreas to get insulin secretion going, because these are signs that blood glucose level will go up. So let's get the insulin secretion going so that we're ready for that glucose when it shows up in the blood. We also have negative feedback mechanisms. So again, this is a picture from another textbook. So our stimulus is an increase in plasma glucose. And the beta cell is essentially our sensor and integrator. So when the beta cell senses high glucose, it releases insulin into the blood. Then insulin goes to insulin-sensitive cells, binds to their receptors, and tells those cells to use and store glucose. If you're using glucose and storing it, then your glucose levels are brought to a healthy range. And you see the same thing in this flow chart. The stimulus is an increase in plasma glucose. The beta cells respond by secreting insulin. Insulin goes up in the blood. It tells your fat cells and muscle to take up glucose and use it. It tells your liver to not make glucose and to store that glucose as glycogen and fat. And if you're using and storing the glucose, then your blood glucose level goes back to healthy levels. So let's make sure we all understand how negative feedback is different than feed forward. In negative feedback, the parameter being controlled is blood glucose level. When blood glucose level goes up, you secrete insulin. Insulin then does things to bring blood glucose back down. So the stimulus is an increase in blood glucose level. The response is a decrease in blood glucose level. That's a classic negative feedback mechanism. Now, feed forward means that blood glucose level, the thing that's being regulated, has not gone up yet. Feed forward means there are other signs that anticipate an increase in blood glucose level. And those two things are parasympathetic stimulation and glucose in the small intestine, leading to the secretion of the incretins, GIP and GLP. Now, insulin does all kinds of wonderful things. It promotes glucose usage and anabolism. So it's going to tell insulin-sensitive cells to take glucose up, phosphorylate it, and use it in glycolysis to make ATP. It's going to promote the uptake of amino acid, and then your muscle and your liver can then make proteins out of it. And remember that insulin-like growth factor and growth hormone are going to help us do that. It's going to increase triglyceride synthesis and the storage of triglycerides in the adipose tissue. And it's going to increase glycogen synthesis and inhibit glycogen breakdown. So once again, we're going to use glucose, we're going to store glucose, and get blood glucose back to where it should be. Also remember that high amino acids stimulate insulin. So we're going to promote amino acid uptake and then taking those amino acids and turning them into proteins. So here's a diagram from your textbook showing you the insulin receptor. And when insulin binds, we get phosphorylation by a tyrosine kinase. So the insulin receptor doesn't use a G protein. It sort of leaves out the middleman and goes right to an enzyme called a tyrosine kinase. Phosphorylation allows it to interact with substances inside the cell. And then there's a big, long intracellular pathway. And basically about 10 little processes. It's like, I had a colleague that described it as tag, you're it. So one molecule talks to the next molecule, and that molecule talks to the next molecule. And it's a big, long pathway, and many, many people are studying the ins and outs of that pathway. One of the things that happens is that GLUT transporters inside the cell are then translocated to the surface so that glucose can come in along a concentration gradient. This is another picture showing that same idea, that here's insulin binding to its receptors. There's a signal transduction pathway, and the major thing that happens is vesicles containing glucose transporters are merged with the cell. Now there's a way for glucose to get in. In the absence of insulin, then these glucose transporters are removed and sent back into the cell. So insulin-sensitive cells have to have insulin in order to get glucose in. If they don't have insulin, then their glucose transporters are not in the membrane, and glucose has no way to get in. All right. Now, how does insulin decrease the blood glucose level? Well, one of the major ways is that it helps cells use glucose by helping the glucose to get into the cell. If a person did not have insulin or could not respond to their insulin, then the cell would have difficulty putting the GLUT transporters in the membrane. Glucose wouldn't be able to come into the cell, and glucose would be stuck out in the plasma, and that would make the blood glucose level really, really high. And that results in a condition called hyperglycemia. So wherever you view this presentation, maybe you're going to listen to it before class, maybe you'll listen to it after class. I hope that you will identify your questions and that you'll put them on the forum so I can help you with the things that you're grappling with. And I bet other people have the same questions you do. So feel free to put those questions on there. Next lecture, we are going to talk about the post-absorptive state. And I am recording this session in the spring of 2017. So this Wednesday, we're also going to do course evaluations, and I encourage you to come to class so you can make your opinion heard. I want to thank you for listening to this presentation. I appreciate how hard you're all working and how enthusiastically you're approaching the topic of physiology. And I look forward to see you in class next time.